Hi, hello. This is the video for the topic 7, the gene. And then in this topic, we have three subtopics. The first one is we call it central dogma. In the common cases, how a DNA sequence become a protein sequence. So in a gene. So firstly, what is a gene? A gene is say a DNA sequence, but not every DNA sequence. A DNA sequence with a product, uh, product, and so in the cases, what kind of product it can be? This can be an RNA or can be a protein. So a gene product cannot be a carbohydrate, right, or lipids. But carbohydrate and lipids were mainly made by RNA and actually mainly by the protein as an enzyme to make carbohydrate and lipids. And also in the whole genome, a genome is the last topic, but let's talk about something in the future, in advance. So a genome means all the DNA, all the different chromosomes in the human body, and plus mitochondrial DNA. So all the DNA, the whole set of DNA in a human cell, we call it a genome. And in genome has all the DNA in the genome, only a small percent of those is actually have a product. And particularly only 1.5% of those are protein coding. Most of those genes are proteins. Some of those are RNAs. We can already talk about later. And then even though most of the genes are coding protein, only 1.5% of the genome has coded proteins. For the rest of the genome, what are function, we'll talk about later also. And and for all of the rest, most of the rest, for example, regulatory system, like the promoter, is not a gene. And then in the genome, the most abundant part is the transposable element. Transposable elements. These are not a gene. So this is a complete definition of a gene. Next, let's say a gene is a DNA sequence. Eventually, some gene become RNA, some gene from protein, but now we focus on how the DNA sequence become a protein sequence. There are two steps. First, the DNA need to become a RNA sequence, and this we call it transcription. And then the RNA sequence become a protein sequence, and this we call it translation. So, and then let's first focus on the transcription. Transcription. Here, there's a sequence. This is a DNA sequence. How a DNA sequence become a RNA sequence? And think about this. We just learned from the DNA replication. A DNA replication is a copy, 1 copy to 2 copy of the whole genome, whole DNA genome. But in here, this is something different because you only pick a small part of the genome to transcribe into RNA. So how come the cells find where to start with? That's something in the upstream of the gene called promoter. Promoter is a DNA sequence upstream of the gene. This is the gene. And this is not a gene because it has no product, but it's essential for a gene to be expressed because without a promoter, a gene without a promoter cannot be expressed at all. And then the promoter is a location where you have some protein to bind to. It's called transcription factors. Transcription factors need to bind to the promoter, and then transcription factors recruits this called RNA polymerase. It recruits RNA polymerase, and then this initiate the the transcription. Once it initiated, the RNA promoter will walk along the gene and based on the gene sequence made the RNA. And this RNA made is called an mRNA. And then next to say, how is the gene is located in the genome? First, again, this is the promoter. This is the promoter. And then this one, the TSS, they call it transcription start site. And then from TSS here, this is the first sequence that really code a protein. And then another one. And this sequence that really code a protein, call it exon, the exon 3. And eventually, that's the end. This is the end. Transcription end site, TES. From TSS to ETS in the genome, this protein coding sequence are scattered in several fragments. Each of fragment called a chromosome. And then between the sequence between the chromosome called intron, between the exons called intron. And this from TES to the first exon, this is called 5' UTR. UTR stands for untranslated region, and this is 3' UTR. So at the beginning, from here to here, everything is made and it is transcribed. And for a mature mRNA, the introns will eventually remove, will remove from the transcript. It only have a continuous protein coding sequence, 5' UTR and 3' UTR. And the protein coding sequence in the center, now we have a new name for it, called it open reading frame, called ORF, open reading frame. And this open reading frame here, in this open reading frame, there are multiple genetic codons. Each genetic codon have 3 nucleotides. And these 3 nucleotides, each 3 nucleotides will recognize by tRNA. tRNA. A tRNA is like kind of L-shaped structure, and then this side is called anticodon. Each anticodon will bind to one of the codon, and then this tRNA work like a translator or bridge, because this side is an amino acid. Each anticodon is related to one specific amino acids. In this cases, this tRNA can translate the genetic codon into amino acids. So next, let's see, how does it work? How does translation work? Now, in translation work in a molecular machine called ribosome. Some of this ribosome, and a ribosome has 2 subunits. The smaller one called small subunits. Small subunits, both of those made of RNA and the protein. And the RNA is more important here in the ribosome than the protein. And the RNA that made of the ribosome called rRNA. And then first, the small subunits capture the mRNA. And then this is called ribosome binding sequence in the 5' UTR of this mRNA. And then in the center, this is the star codon. This is star codon here. And then the first tRNA bind to the first codon, whatever. The first amino acid is coming here. And then after the first amino acid coming here, now we have the large units binding to this complex. Now you have the full complex of the ribosome translation machine. And in the large units, there are 3 slots for the tRNA. This one is called, the center one is called P. And this is called P because this is the one you have the polypeptide. And this is called A. This is called A because here the tRNA at this point carrying amino acids. And this is called E. The E probably because this is the position that the tRNA escape or existing in the machine, or the tRNA here carry an empty tRNA. So now this A, at first, this A is empty. Any tRNA will come, but only if when this tRNA match the codon right here. When the tRNA come here, match the codon here, it trigger the machine to and this tRNA carry amino acids, and this amino acids added to the amino acids, or in the most cases now here is a polypeptide, add to this add to the polypeptide or the amino acids in the P position. And when this happens, this form the peptide bond. This is how the peptide bond is found. It's done in the large units. And when this peptide is done, this large units, the small units, both moving forward. Now here, this coding, this start codon become into the E position, and this tRNA goes to E position. Next, once the E position, will next will be getting out, and then this one go to the previous tRNA in A position will go to the P position, and then you have the A position being empty. When the A position is empty, new one will come again to the A position, and then the amino acid carry the new tRNA in A position will further added to the polypeptides in the P position. Actually, the whole polypeptide now move to the A position, and then they keep moving this word. And since everything moving, the ribosome moving to the 3' of the mRNA, and the mRNA moving to the 5' itself. Anyway, so and then this tRNA, the empty tRNA, is squeezed out from the E sites, E position. And then eventually now at the end, you have the stop codon here. And what happened to the stop codon? Stop codon does not match to any tRNA, but rather a protein similar to a tRNA coming to here. And this protein will trigger the disassembly of everything. Then you have the mRNA will gone. mRNA will bind to another ribosomes. And actually, the mRNA can be used several times. And then at the same time, you have a polypeptide, and this polypeptide will be coded to a protein. And then here, next, let's talk about one more thing is called the regulator. The whole process is called gene expression. And then gene expression is very important. And we human have about 20 to 25,000 genes in the whole genome, but in each single cells at each specific time point, only a subset of all of the genes are going to be expressed. Next, we talk about how this is regulated. First, the gene exists, the DNA exists in the chromatin. In most cases, the chromatin is closed. Closed chromatin need to be open, to open chromatin. And then how you can open the chromatin? There are some ways to open the chromatin. The key thing here is a DNA wrapping on the surface of a histone. And if you add an acetyl group into the histone, it open up. It's open. This open up the chromatin. So histone acetylation open up the chromatin, and open up the chromatin increased the gene increased the gene expression. So this is one way to increase gene expression. And also the gene expression is regulated by heavily based promoter. Some gene expressing everywhere is we call it home housekeeping genes. Some genes are only specifically expressed in some certain cells, and how they happen, it largely depends on their promoter. A promoter determines when and where a gene expressed, because when and where a gene need to be expressed only when a transcription factor binds to the promoter, and transcription factor is highly regulated as when and where. And then it's very complicated because transcription factor is also a protein, also a gene. So what regulate transcription factor is very complicated. It's a network. We don't have a clear picture yet, but I hope in the near future we can have it. And then sometimes, again, this is a promoter. Sometimes a promoter cannot be bound by a transcription factor. One is heavily methylated. If you remember methylation is a CpG, and C has a methylation here. So if a promoter has a methylated, it is hard for transcription factor to bind to it, and this is called a promoter methylation. Methylation will suppress gene expression. And then, so you say everything worked fine. Now you get an mRNA, and mRNA into the cytoplasm, and then here, mRNA has a 5' cap. This is the and then a poly A tail. This is the processing mRNA. It always has a 5' cap and poly A tail. This is important because mRNA without a poly A tail or 5' cap will be degraded very fast in the cytoplasm. This is a cell protection mechanism. Any mRNA without this protection will be degraded. And then this mRNA, when it get into the cells, it can go several rounds of translation. And each round of translation, the mRNA becomes less stable. And after several rounds, this mRNA will degrade by itself. However, if you something else, call it, if this tiny small mRNA, call it microRNA. MicroRNA is about 21 to 23 base, not base pair, base. And if this mRNA happen to be captured by the microRNA by a complementary binding to the center, this microRNA will make the poly A tail to be removed, shortening much faster. And this mRNA become very unstable. So mRNA binding silence the mRNA. So there's another level of regulation of gene expression. Yeah. So finally, even though you have a protein here, there's another mechanism called ubiquitination. Ubiquitination means you have a you have multiple ubiquitin labeled to a protein, and once a protein is labeled by this ubiquitin, it's degraded like paper shredding machines. And this labeling is so when a protein is no longer needed, for example, cells made an emergency protein, may some proteins trigger the absorption of the glucose, and then you do not have enough glucose, you don't want the protein to be exist anymore. You use ubiquitination to label the protein. This is called a post-translational regulation of the gene expression. And here in this we make a special example called HIF1 alpha. HIF1 alpha is a master transcription factor. And once HIF alpha is activated, the cells steer all this direction to use less oxygen, because this HIF alpha is the emergency protein to survive the cell through a hypoxia condition. And in this case, HIF alpha is keep being ubiquitinated and degraded when the cells have enough oxygen. And then for a fast response for the hypoxic condition, Whenever there's no oxidation, Higgs alpha no longer get degraded, and then this Higgs alpha stay to help the cell survive in a condition that in a hypoxic condition. So lastly, I'm not sure I mentioned this. So lastly, we talked about genome. We already mentioned that only 1.5% of the genome is the coding sequence. And then here we mentioned, let's say, particularly the transposable elements. Transposable elements actually came from the virus. Some virus, nowadays, there's still some, this like HIV. HIV is this type of virus. When this virus infects the human cells, they integrate their genome, this is human genome and the virus. They integrate that genome into the human genome. And then some of those transposable elements not only integrate those genome here and then make a copy. They copy itself to amplify multiple copy in the human genome. And then because they can amplify, just like a virus, amplify themselves in the genome, about 50% of the whole genome actually is occupied by these transposable elements. And they are still called transposable elements, but nowadays most of those can no longer actively transpose themselves from one places to another places. So nowadays our genome are quite stable. And in the history, they do contribute to something called gene duplication. So I have a picture, I have an illustration in the slides you can read, but keep in mind, if these two duplication, it will lead to the unequal crossing over. And unequal crossing over will cause a gene duplication. One of the chromosomes carry two copies of the same gene. And this, when you have two copies of the same gene, think about it. One gene can be in charge of the old function of this gene, and the other gene can induce or can accumulate more DNA mutation. And then at the beginning, the DNA mutation will lead to nothing. But when you accumulate more mutation, a new gene may arise, have a new function. This is a major way the human genome can evolve to have more and more new genes. In fact, most of our genes that currently exist in the human genome are a family. All of those families depend on are the consequence of a gene duplication. So yeah, that's it. That's all for this topic. Have fun and see you next time.